I'm going to move straight to our next speaker, which is Dr. Leonie Suter, who is going to talk to us on using environmental DNA to survey Antarctic krill. Leonie completed her PhD in Switzerland, studying how plants can adapt to a changing climate. She then sailed on a small yacht from the high Arctic to the Caribbean for 2 years with her husband and her dog, and ended up studying the sex life of Antarctic krill in Tasmania. What a journey. Leonie's current work focuses on how we can detect life in the Southern Ocean using environmental DNA. Highly topical area. Thanks, Leonie.
Thank you. So I'll jump straight in. What is environmental DNA? Well, environmental DNA is the DNA that's released by any organism at any time into any environment. In the case of Antarctic krill, that might be molds or feces or eggs or any dead decaying tissue that's released into the environment and then spreads in that environment. Now, any other organism in the marine environment is doing the same thing, so that we end up with a soup of DNA of all the surrounding organisms in the water, in every litre of seawater. We can access that information by taking a small water sample from the environment, and that can be done, for example, from directly from a ship, or anywhere we really can get to, from under sea ice or even from the deep sea. And this information can then be used to describe communities, community composition, species distribution, how species interact with each other. And over time, we can monitor the response of these species and communities to change. And importantly, we can do that without ever having seen any of these species in person, so to say. We just use the traces they leave behind, the DNA signatures. Now, we can describe whole communities, but we can also zoom in and look at single species in more detail. And that's what we've been doing here. I don't really need to say anymore why krill. I think you've heard enough about why krill. It's an incredibly important keystone species in the Southern Ocean, really abundant, and a lot of predators depend on it. It's also targeted by the krill fisheries, and for the sustainable management of the krill fisheries, we need to know how much krill there is and what its distribution is. You've already seen about the amazing tools we have for acoustic surveys. We also use trawl surveys to answer these questions. But here we investigated whether eDNA-based methods can be additional tools to survey where Antarctic krill can be found and get insights about their habitat use and abundance. Let me go back one. There. What we don't know about eDNA in the Southern Ocean is really for how long it sticks around. So once it's been released into the environment, the DNA strings actually begin to break, and they get smaller and smaller and more fragmented, and once they're really short, we can't really tell who it belongs to anymore. So there's a limit to for how long we can detect it, but we don't really understand what that is. To understand more about that, we set up an experiment in the krill lab at the AAD in Kingston, and we put some krill into some small tanks, and we let them release their eDNA into those tanks for 2 days, and then we removed the krill, and we sampled those tanks for 2 weeks to see how the eDNA of krill decays over that time. Then we did our genetic tests, and in fact we used genetic markers of different lengths to not only see how the DNA might decay over time, but also whether it fragments more over time, so we can get an idea for how long the eDNA has been floating around. And this is what we found. So you can see that over the course of these 14 days, the DNA degrades quite rapidly, but also, so we have these 3 different length markers. Over the course of these 2 weeks, the DNA begins to fragment more and more. So based on this fragmentation, we then get an idea of whether the eDNA has been released recently into the environment or has been floating around for a longer time, which will help us to interpret the data we're actually collecting. We've then taken this method to the field. This is a resupply voyage to Davis Station, where on the way back we collected small water samples all the way to Hobart, and we measured the Antarctic krill DNA from Davis Station to Hobart, and the size of those circles indicate how much we actually found for each location. But we can also get an idea now how fresh or old this eDNA is. So the white ones contain eDNA that's less fragmented, whereas the dark ones contain more fragmented eDNA, so it's probably been floating around for a longer time. This is just one of the markers we can use to get information out of these samples. We can also target other species. For example, we used a marker that can detect mammals, and in the very same samples, we found a whole range of whale and seal species. So depending on what you're interested in, you can target your markers and find really anything you like. The next steps are going to be to actually investigate areas that are usually a lot more difficult to survey. For example, we know that krill are using the deep sea occasionally, probably, as a habitat, but we don't really know how often, how widespread. We've got samples from the deep sea that we can use with this method to see whether they're commonly found there or whether it's just every now and then. We want to compare this new method to already existing methods to understand better what this data actually means, and then we want to use a whole range of additional markers, such as markers targeting predator eDNA or overall animal communities, to understand better how krill fits into the Southern Ocean ecosystem. That's it. Thank you.